In this lesson, we're going to look at quite a complex topic. Today, we are looking at what are monoclonal antibodies and how are they produced. This links to the specification point, describe the production of monoclonal antibodies, including the use of lymphocytes, which produce desired antibodies but do not divide, produce of hybridoma cells, and hybridoma cells producing antibodies as they divide. To start off with, we need to get some basic definitions down, and the first one I want to look at is that of an antibody. Antibodies are proteins, and the point of them is they neutralize, so they stop pathogens in a number of different ways, and we don't need to worry about the number of different ways just yet. What we can see here is an antibody that's been secreted, so that means given out by a white blood cell, usually called a B cell. And this antibody latches on to something called an antigen on the surface of the pathogen, and as it latches on, it flags it to the white blood cells to come and destroy. Step 1, you vaccinate a mouse. So let's say you wanted to produce antibodies to destroy the pathogen tetanus. You would inject the mouse with a shot of tetanus, so we've got a live vaccine there. The mouse would start to produce a big immune response to it, and as a result start knocking out antibodies. These antibodies we're going to harvest, and that leads us to step 2. Step 2 is a rather unfortunate end to the mouse. You amputate, so you remove the spleen. The spleen is where a lot of the antibodies are produced. Take it out, and then we have a live source of antibodies for the next stage. Step 3 is isolating the myeloma cell. Now, myeloma cells are just cancer, and cancer can be hugely damaging, as you're well aware. It does have the property of producing many identical cells very, very quickly. So essentially, the myeloma cell clones itself reliably time and time again. This is a really useful property when it comes to monoclonal antibodies. Step 4 is the really clever part. Some really nice science goes on here. You take the B cell, and remember these B cells produce a huge amount of antibodies, and you fuse them, so you join them with the cancer cell, the myeloma cell. This creates a hybrid cell called a hybridoma. Now the hybridoma cell takes the property of the B cell, i.e. it produces a lot of antibodies, the property of the myeloma cell, it divides very quickly and starts to be producing a large amount of antibodies because it's continually dividing, and as it divides it produces these antibodies. Step 5, you incubate these cells, so the hybridoma cells, you incubate them and you collect the antibodies that they produce, and they'll keep producing antibodies because the cancer just keeps dividing.